Good afternoon, guests, educators, and students. My name is Warren Nelson, and I'm here to introduce my fellow researchers, which consist of me, Crystal, Elle, Megan, and Becca. The area of Silver Falls State Park designated CAT 2.7, we wanted to know how the richness and abundance in the fungi in the burned area compared to unburned area. Our fungi research focused on answering two questions instead of one because we had to pivot our research halfway through due to early snowfall and interrupting our original plan. Our second question was how does the fungus richness and abundance change depending on the elevation of Silver Falls State Park? It does in the first research question because fires are becoming more common when they do happen, here worse than they have been. We wanted to see if the fires will impact the growth of fungi. Silver Falls State Park was partially burned in the 2020 Beach Creek fire. We decided to compare an area along the Cameron Trail that was known as CAT 2.7 that was burned by a fire to an area along the same trail where the fire did not reach. We have pivoted our research. We decided to look at elevation. Our unburned CAT 2.7 location was approximately 2,000 feet. We selected a spot along the North Perimeter Trail that was approximately 1,500 feet and a spot at the base of North Falls that was approximately 1,100 feet. Our hypothesis was that the burned part of the forest would have fewer fungi due to the fire eliminating visible fungi, which hold spores, and the loss of them would limit reproduction. We thought the majority of fungi found in the burned area would be conch because they grow on dead wood. We believe the unburned forest would have more species, richness, and abundance. We hypothesized that the fungi would be more abundant in the rich in the medium elevation than the high and low, because it's less extreme in weather conditions. Fungi are an important part of the habitat. They help decompose dead organisms and feed all the plants around them. In addition, many fungi are mycorrhizal, which means they share a special symbiotic relationship with trees. The hyphae grow around the tree roots and are mutually beneficial. Species of mycorrhizal also help trees regulate CO2, which can help fight climate change. A fire is lethal to the fungi on tree roots. If the soil reaches a temperature around 60 degrees Celsius during a fire, the soil can reach temperatures of 600 degrees Celsius. This means everything above the surface is mostly burned or dead.
Fungi were initially classified as plants by Carl Linnaeus in the 18th century. He classified living organisms into two categories, plants and animals. In 1969, Robert Lydekker proposed the 5-human taxonomic classification: Animalia, Plantae, Fungi, Platista, and Monera. Ongoing research has shown that fungi are more closely related to animals than plants. The Beach Creek fire happened on August 16, 2020, when lightning struck near the Opal Creek Ancient Forest Center. A record windstorm fueled the flames, causing them to envelop their surroundings. Thankfully, a change in wind direction prevented greater damage. However, 5 people died from it, homes were lost, and almost 200,000 acres of land were burned, including part of Silver Falls State Park. Fungi live worldwide, including deserts, aquatic habitats, and terrestrial environments. However, most fungi grow on soil or dead matter since they decompose in organisms. About 80% of fungi are associated with trees. Many fungi thrive best at 12 to 30 degrees Celsius, around 54 to 86 degrees Fahrenheit. Heterotrophs, or organisms that eat other animals or plants for nutrients to survive. Autotrophs are producers that make their food from raw materials or energy. Fungi are heterotrophs and differ from plants, which are autotrophs. Plants have structures which transport water and other minerals from the soil to the plant, and then move sucrose and amino acids down from the leaves. Fungi obtain their food from outside themselves. Many different animals eat fungi. Some fungi are parasitic and can eat each other as well. Lastly, humans eat fungi, especially mushrooms. They are used to make bread and ferment food and drinks, as well as in medicine. Fungi play an important role in the complex food web that helps ecosystems every day. Fungi reproduce asexually or sexually by producing spores that disperse from the parent fungi. These light, small spores float in the air and around 1 trillion spores are released. In sexual reproduction, the spores disperse until they find a livable environment. Then, they grow grow a root-like structure called mycelium. This will spread underground in a hyphal mound. It will then form a fruit body, which starts as a pinhead. This grows into the mushroom fruiting body, and the cycle begins again. The asexual life cycle begins with a spore that will settle to build its mycelium. Some fungal species can only reproduce a certain way. If conditions are adverse, some fungus may opt for sexual reproduction to adapt better. When fungi reproduce asexually, mitospores are produced that are identical to the parent fungus. They later grow in a new set of mycelium, and the life cycle repeats. Many nonprofit organizations and conservation groups are working to protect fungi. One example is the Fungus Conservation Pledge. This initiative aims to prioritize fungi within global biodiversity agreements. The United Kingdom and Chile presented this aim to intensify fungi protection at the COP16 conference on October of 2024. The UK and Chile are working together to protect fungi. Chile's Environment Minister, Maisa Rojas, said, "The purpose of this agreement is clear: to incorporate fungi into global conservation strategies and frameworks, highlighting the key role they play in combating climate change, biodiversity loss, and promoting sustainable economic development."
The materials that we used in the field were identification books, pens, study paper, note cards, transect, a picture-taking device, and field trip forms. Questions like, where are you going? What are you doing? What's the weather? What time are you starting? And what does the site look like? The materials that we used in the classroom were identification books, computers, the digital photos that we took, and spore prints. All our research was located at Silver Falls State Park. On the first trip on October 28th, 2024, we went to the Catamount Trail at mile 2.7. On our second trip, November 11th, we had to turn back due to high amounts of snowfall. On our third trip, December 9th, we visited North Falls and Winter Falls. After we arrived at Silver Falls State Park, we grabbed the research materials. We walked along the site until we found our research location. We found a location with walkable terrain. We made sure that there was fungus in the area. We then laid out a transect in a straight line perpendicular to the trail for around 30 feet. We split the people into two groups, one on the right of the transect and one on the left of the transect. We only went 5 feet to either side of the transect. Both groups stuck to their side of the transect and looked for fungi. Once we saw fungus, we took a photo of it, picked it up carefully, wrote down the letter of the group we were in, the observation, and the number on the note card. We took a picture of the fungus right next to the number on the note card. We recorded the number and letter of the mushroom on the paper. We wrote the characteristics of the fungus, like color, texture, gills or pores, and other details. We also tried to identify the type of fungi. We kept doing this while slowly making our way to the end of the transect. We measured richness by the amount of unique genera. We measured abundance by the number of observations of fungi we made. We counted one observation as each patch of fungus, the same species that we found close together. If we found more of the species some distance away, we counted it as a new observation. After we finished, we repeated this procedure at our next location. Once we finished everything, we took the materials and some mushrooms we saved for spore prints back to the van. Once we got back to the school, we grabbed the identification books and our Chromebooks. We then made a digital data sheet, typed in the number and descriptions of the fungi. We also used the book to identify the fungi and put that in the sheet too.
On October 28th, our group started research on the effect a burn habitat has on the abundance and richness of the fungus in the environment. We found 20 observations of fungus in the burned area and 21 observations of fungi in our unburned area. On November 28th, our group redirected our research towards the correlation of the abundance and richness of fungus in the elevations grown out. Of the 28 observations of fungus in the burned area, the most populous genus was Mycena, of which we observed 14. There were 4 Foliota, 2 each of Tremella and Navatellia, 1 each of Cordinarius, Hyphloma, Lactarius, Mold, Sterum, and Trametes. We are unable to identify
genus of the mold, which is why we left its description as just mold. In the unburned area, we found a total of 21 observations. Mycena was again the most common with 7 observations. There were 4 Rusula, 2 Atheniella, 2 Anusi, 2 Tremella, and 1 each of Hebeloma, Ephiota, Soleus, and Tremidis. We used this data for our high elevation sample. We found a total of 18 observations of fungus in the North Falls medium elevation. There were 5 each of Lactarius and Mycena. There were 2 Pseudodinahitum, and only 1 each of Grisette, Clavulina, Hymengastrius, Fomitopsis, and Gumpinis. There were a total of 13 observations of fungus at the North Falls low elevation. There were 2 each of Fomitopsis, Lactarius, Mycena. There were 1 each of Bolete, Cavlina, Cordinaris, Galerina, Grisette, Nidulariaceae, and Tremella varinis. In the burned area, there was a higher abundance compared to the unburned, with 28 observations in the burned and 21 in the unburned. The richness in both the burned and the unburned areas were pretty evenly distributed, with 10 and 9 respectively. The high and medium elevations had the same abundance. The lower elevations abundance is lower than the other 2 areas. The high and medium elevations richness is the same, and the low elevation is a little higher, but still very similar. When we compare our hypothesis to our data, we found that our hypothesis was incorrect. We originally thought there would be more fungus in the unburned than most of the burned, which our data does not support. Also, there was more unique genera in the burned compared to the unburned. The abundance was tied in the medium and high elevation and somewhat lower when it came to low elevation.
There was a larger abundance of 28 in a burned area than an unburned abundance of 21. It's possible that this happened because there would be abundant dead organic matter left over from the fire, providing energy sources for saprophytic fungi. The burned area has a higher richness than the unburned by 1. It might be that fire doesn't make forests uninhabitable for fungi. Our data shows that though some genera did fully disappear, new ones appeared from the drastic change in environment. An example of this is Pholiota highlandensis, which is a type of fungi that only appears in forests after fire. Fungi like those could be why abundance and richness increase after fires. It's also backed up by Devenhart's idea that the difference in richness before and after fires was decreased to no change. Following this theory, 5 years may have simply been enough years post-fire for that to happen. There is a larger abundance in the medium and high elevations than in the low elevation. It could be that the particular forest we used for low elevation was unrelated to the elevation, a poor place for fungi. That particular transect was also set in a trail loop, rather than extending outwards. It would have had more human interference as a result, since it's a more popular and easily accessible trail compared to the perimeter trail. It's important to note the low elevation was 400 feet beneath medium elevation, and medium elevation was 500 feet below high elevation. Despite this difference, we found that medium and high elevation have the same abundance of fungi, implying that the 400-foot difference between medium and low elevations was more impactful than the 500-foot difference between medium and high. The richness is the same for medium and high elevations, but higher for low elevation, though it is not a statistically significant difference. Because of this, it would seem that the number of genera remains relatively equal across different elevations, but the amount of fungi doesn't, suggesting that abundance is much more affected by elevation than richness is. Uso, Kenwright, and Burns discovered that heat treatment causes some fungi to increase in population while others decrease. It does not cause one specific thing to happen to the resistant propagule community of ectomycorrhizal fungi. A wild forest fire is a bit different than controlled heating of soil, but we had similar results because the fire didn't affect all the genera in one way. Rusula was significantly lower in the burned area. It was the second most populous fungi in unburned and completely missing from the burned. Most genera decreased in population, but Mycena and previously absent fungi increased. As our research question is new to JGEMS, the first thing we would do to continue this project is take more data. We would start by repeating our study at Silver Falls to see if it continues to match our initial data. Then, we would compare those findings to newly burned forests of the same elevation to see how it compares. Afterward, we would compare data from burned recovering forests of different elevation to see if that reflects recovery rates. On the October 28th field trip, we brought normal paper instead of Write in the Rain paper, which caused almost illegible writing at times. On that same day, there was a miscommunication on what key opened the trail gate, which impacted our collection time. The heavy snowfall during the November 18th trip was a considerable and unforeseen limitation to our research. The weather conditions forced us to abandon the data collection, and we were with a stuck van and no fungi in sight. As middle school students, we only had little time and field trips at our disposal, which means we have a very small sample size. Consequently, there are many possibilities for what our data means, but not as many precise and evidence-based explanations. If this were to be continued in the next year, it might fix some of those problems because we would have more data. To make sure certain limitations don't occur again, it would be wise to start earlier in the year. It might be more beneficial to keep it to one question. If the next group were to go earlier in the year and have no problems with snow, they wouldn't have to split their focus. A question we considered ourselves was how the growth of fungi in burned forests near logging trails, which would have been cleared and aided with human hand, compared with fungi growing in a recovering forest without. It would be an interesting project and pertinent to modern struggles. A special thanks to Mrs. Ames, Miss Izzy, Mrs. Olson, Joe Hershenko, Mr. Whittle, Mrs. Whittle, and the panelists. Any questions?
All right, panelists?
So you were looking at altitude as a variable, and you were looking at burned and unburned as a variable. And if I recall, you had 3 sites, which was the CAT 2.7, which was a burned area, the Newt Loop, which was the mid-altitude unburned, and then the lower altitude was the canyon.
Well, the high elevation, the data we used for high elevation was actually the unburned data. We took up high elevation to make sure it was all unburned. But yes.
And no, but you did do comparisons of burned and unburned, and that was within the CAT 2.7 area when you were chasing. Is that correct? Okay. Yeah, I really like that you were considering just some environmental differences, because the canyon is quite a bit different in what's growing in there than at the other altitude elevation. So, considering altitude, but also the impact of altitude and terrain in doing those comparisons. I jotted down a question, you answered that in the course of saying these things. So that's my question. Or observation.
Becca, Dagan, L, Crystal, Oren, awesome job. I really love fungi, so I was really excited to hear your presentation. And Silver Falls is a beautiful place, so lucky that you got to go there a couple times. Question, do you think fires are good for that particular ecosystem over there?
I think that fires, sometimes they're good, but sometimes they can destroy homes, and sometimes people can die too because of them. However, they are good sometimes for the ecosystem.
In addition to that, the forest, it makes an overabundance of fungi, and we like fungi, but that's mostly because in this case, it's helping the forest regrow. So it would be not a good idea to have more forests, so we have more fungi.
Yeah, maybe I should have rephrased it as, is that an ecosystem that is normal to fires as part of the development of that forest? Do you know? It's okay if you don't.
why it's so cool, like anybody that took that away. Like why are they so special? Because I know a lot of people like them, but I don't really know why. I like animals, so I'm like, heck.
So for me, something I didn't know a lot about was how they reproduce, like asexual reproduction and sexual reproduction. And so that was something really interesting that I learned.
I found it really cool that they are more closely related to animals than plants.
I found it interesting how the different ways they help forest recover, because you first thought they decompose the dead organisms, it helps the forest, but there are also other fungi besides saprophytic that help the mycorrhizal of the trees. And the mycorrhizal especially help with CO2, which stops them from happening in the first place. I thought it was really interesting.
I found all the types of like parasitic fungus very interesting. I found researching that at home was really a fun thing to look through.
The trees, how it helps out trees and all the plants around it.
Seeing, as I recall in your results, fewer, at the lower elevation in the canyon, you saw lower diversity, is that correct, or lower numbers? Numbers of. What would you say observationally, about that area, maybe even in terms of the food source, things like that? If it's maybe not directly altitude, but the environment that they were in that was affected, why would you speculate? Did you observe anything that would lead you to believe maybe this is why there's fewer mushrooms here?
When we went to the burn site, in my opinion, I thought it made sense that there would be a higher abundance of the fungus. Because more dead things, I just assumed they'd be able to latch onto more. And then when we got to the unburned, just my fruitions were fed, I believe. That just really helped out with my opinion on that.
In addition, the low elevation again was a very popular trail for us people. It's part of the 10 waterfall trail, and as people step on the trail and they walk it, it makes the soil more compact, which does not help with for fungi growing there. It prevents them.
Are mycena just particularly common in Oregon, or is there something about that area that they really like to grow?
Are mycena common?
The mycena are common, so when we studied that, we found that in generally the mycena doesn't mean anything's particularly bad or that forest fire makes something happen to make mycena grow. It's just an indicator of a normal healthy forest.
What do those mycena look like for people who might not know what a mycena is? For the most part. What did the most of the mycena look like?
Did we put an image in the presentation?
Just tell me, what are they? What did we call them?
A three-letter.
LBM. Little brown mushroom.
So what I'm trying to get them to say is that all of these little tiny mushrooms, you go out and you see a bunch of little brown mushrooms, there's a very high likelihood that all that giant patch of little brown mushrooms are mycena. So they're just extremely common basically everywhere. We've got about 2 more minutes. If there's any other panel questions or Mr. Weddle or our audience questions, former student questions.
Was the only book that was used as point of reference the fungus or mushrooms demystified by David Rohrer, or were there other researchers or scientists in the field of fungus?
There was definitely more books than that. There was, for example, All the Rain Promises More, also written by David Aurora.
Mushrooms off Cascadia was a big one. It focus specifically in the Salem, Post area. And also Joe.
One more? Anybody? Mr. Weddle?
So cordyceps fungus have been in the news and the media. Do we have any cordyceps fungus here, and what are they?
I can answer the what are they?
Okay.
They are a type of parasitic fungus that tend to take over, I'm pretty sure just ants in general. It might just be a certain type, but it'll take over a plant's ant's brain, and then I'm pretty sure it'll kill itself.
Do we have them here?
I am pretty sure it did.
I don't know the answer to that.
Can anybody confirm?
I'm fairly certain we don't have
The Last of Us.
I think there's something
I
Do
don't
we
think-
have any cordyceps?
I don't think it grows here, although I can say that the Green Salmon Cafe in Yachats does sell a cookie that uses cortisones in the ingredient list.
Wow. Don't eat
Wild.
that.
No, it's actually, it's quite good, it's done, it's not a living one. And also the cordyceps, I haven't been a fan of the show, but I did love the
I
first
didn't either,
scene, where there was a discussion that the cordyceps cannot survive in the human body temperature range. But it suggests that perhaps, the talk taking place in the 1960s, perhaps if there's environmental change like a 2-degree warming of the planet, it might adapt.
So look out.
All right, one final round of applause for fungus.